So this is a work we started 4 years ago with support from FARA initially to find or discover biomarkers of disease progression in Friedreich's ataxia using MR spectroscopy, MRI, and diffusion tensor imaging. As you know, spinal cord degeneration occurs early in FRDA. We previously reported the first cross-sectional spectroscopy and diffusion MRI data in the spinal cord in Friedreich's ataxia. And so our goal now was to establish longitudinal progression of spinal cord MRI, MRS findings to identify biomarkers for clinical trials. So here's an overview of what I'm going to show. I'm going to show brain diffusion tensor imaging data. We also have VBM, voxel-based morphometry, but I'm not going to show this today because we have not finished data analysis. And then in the spine, I'm going to show DTI, MR spectroscopy, and morphometry by measuring the cross-sectional area. And at the end, I'm going to show a few slides on pilot data in the dentate nucleus and the dorsal root ganglia. The recruitment, we had 28 subjects with Friedreich's ataxia. The FAST scores was around 40 on average, so it's a relatively early stage cohort. Disease duration was 5.6 years on average. Time from diagnosis, 2.3 years. And we had 20 control subjects that were more or less age and gender match. For longitudinal follow-up, we had 21 patients coming 1 year later for visit 2, and 15 patients so far coming back for visit 3 at 24 months. And we just started recruitment for a 4th year with 5 patients so far. So today I'm going to show the first 3 years of data. The time between V1 and V2 was a little more than a year, and same thing for time between V2 and V3. We have a few data sets excluded, most often due to movement, creating artifact in the images. But we also had one subject who had braces, and that caused artifact in the images. So from that point on, we added that as an exclusion criteria, braces. The methods, all the work was done at 3T. Brain DTI was 1.8 cubic millimeter isotropic with 128 diffusion and 15 B0 and a multi-band factor of 3. Spine morphometry was done with regular T1 weighted images, 1 millimeter cubic isotropic. The spine DTI was done at the C4, C5 level with a resolved sequence, 30 slices and 30 diffusion gradients, 6 B0, and 2 opposite phase encodes to correct for distortions. And finally, the spine spectroscopy was done in the 1.4 milliliter voxel with a semi-laser sequence, echo time 28 milliseconds, and 256 acquisitions, and using LC model to analyze the spectra using water as an internal reference. So I'm going to start with the brain DTI. I want to emphasize the importance of the atlas, and particularly for this study, we created 2 separate atlases, one for control and one for patients. So and then we did TBSS to assess the differences between controls and patients. So in diffusion imaging, we measure a parameter called FA, fractional anisotropy, which measures the degree of order of water diffusion. So very high FA is between 0 and 1, and FA close to 1 means a lot of order, for example, along the fibers, and FA close to 0 means random diffusion. And so when we have axonal loss, for example, we have FA that usually decreases, and when you have demyelination, you also have FA decreasing, but also RD increasing. However, there are some special cases where you can have neural loss of fibers, but FA, instead of decreasing, will increase. For example, if you have fibers that cross and you lose, so fibers that cross would give more directions of diffusion, so you have a low FA. And then if you lose this track, suddenly you have much more order and FA will increase instead of decreasing. So you will see this in some of the images that I will show you now. These are cross-sectional data, differences between patients and controls. So you see, for example, the cortico-spinal tract and differences in motor cortex. There are also differences in the optic radiations in the visual cortex, and a little bit in the hippocampus and amygdala. And here, at different locations in the brain, you can see very well the tract around the dentate nucleus in the cerebellum, and a lot of differences in the midbrain. And here is one example of what I was telling you. So the blue are actually negative changes, which means actually increase in FA, and the orange are decrease in FA. So there are some regions here where the FA is actually increasing. Okay, so now longitudinal data. FA is increasing, especially in the brainstem and the thalamus and the corticospinal tracts here. And FA is decreasing in the posterior limbic internal capsule and corpus callosum. So that was for the brain. Now I'm going to move to spine DTI. For spine DTI, we use two different processing methods, one manual, one automatic. These are the results for manual segmentation at C4, C5. We saw a decrease in fractional anisotropy. In cross-sectional data, it was about 15% lower in patients than in controls. And in longitudinal data over 12 and 24 months, we had a noticeable decrease that reached 9% at 24 months. The same was true for mean diffusivity, that was increasing. And mean diffusivity was 15% higher in patients than in controls. And similarly, in longitudinal, we had an increase in MD over 12 and 24 months. And both the actual and the radial diffusivity were increased. Now, the downside of manual segmentation is very time-consuming, so it's hard to do at different levels of the curve. So we also did a method using automatic segmentation using a tool called Spinal Cord Toolbox, developed in Montreal. And you can see here different levels of the cord segmented automatically. And using this toolbox, you can also determine calculate FA in different subregions of the spinal cord. So you see, for example, here, gray matter and white matter tracks. So it's not very precise because the diffusion images don't have as good resolution as the anatomical images, but we can still get approximate data for different tissues within the cord. The observed changes were similar between manual and automatic analysis. So you see an example here with MD, mean diffusivity. Manual data, automatic data, both show the progression of MD over time with similar magnitude. The changes that we observed were also similar at different levels of the spinal cord. So if we look at C3, C4, C5, C6, you find similar differences between controls and patients, for example, and they are all statistically significant with a P lower than 10⁻⁴ at every level. And as I was telling you, we can look at different subregions of the spinal cord. So here we have FA in white matter, in gray matter, and in dorsal columns. And you can see the decrease in dorsal columns is larger, for example, than in gray matter, which is consistent with what we know of the disease. The next modality is spine spectroscopy. And in spine spectroscopy, the differences between controls and patients are so large that it can be seen in individual subjects. So you can see here a control spectrum and FRDA spectrum. You can see the NA is decreased and myocytol is increased in patients compared to controls. And this is quite remarkable because it's a very small volume. The spectra are quite noisy, but because the differences are so large, we see them in individual patients relatively easily. These are the cross-sectional data, where on average, NA was almost 40% lower in patients than in controls. Myocytol was 30% higher. And when you take the ratio to kind of amplify the differences, there's one going up and the other one going down, you get ratio NA over myocytol almost half in Friedreich's compared to controls. Now, in longitudinal data, we also observe a decrease over time of the ratio NA over myocytol, which reaches 15% over 24 months. And these are the changes of individual subjects relative to baseline over 24 months, and almost all of them decrease over 24 months. Spinal cord morphometry. So for this one, we use regular T1-weighted brain images, and we look at the cervical spine. And because the sensitivity was dropping away from the brain, we chose to do it at C2, C3 level. So we had enough signal. And we use a tool developed in Brazil by Pierre-Gilles et al. Called SpineSight, which allows to manually draw segmentation around the spine by placing points around it. It also allows resampling so we can measure the cross-section perpendicular to the spine. So what we found in cross-sectional was that the area was decreased in Friedreich's compared to controls, 28% lower, with a very high significance. And the eccentricity was 13% higher, so that means the spinal cord becomes flatter, the ellipse becomes more elongated. And that's also consistent with the degeneration of dorsal columns. And in longitudinal, we found a clear decrease over time at 12 months and 24 months, with 18% decrease at 24 months compared to baseline. And these are the changes in individual subjects at 12 and 24 months. You see all of them are decreasing over time. So we look a little bit more at the effect size of the different measures. And I have to put a caveat here that our cohort is still pretty small, so the estimation of effect size is not extremely precise. But on our data, we find that the spine morphometry has the highest effect size at -1 at 12 months and -2 at 24 months, which is actually pretty high. And the spectroscopy and the spine diffusion have effect size around 0.3, 0.4 at 12 months and 0.8 to 1 at 24 months. To calculate the effect size, we took, for each patient, we took the difference of the value at 24 months, for example, minus baseline, and then we took the average of the differences divided by the SD of the differences. So our early stage cohort shows that spine morphometry appears to give the best sensitivity to disease progression, but effect sizes will need to be confirmed in larger cohort in a multi-site setting. And one point I want to emphasize here is even though the spine morphometry gives the best effect size, the different modalities give different and complementary information, so not only on anatomy, but also on neurochemistry and microstructure. And in addition, they can give information at different stages of the disease. So here I did something where I extended the rate of progression toward the past to see, making the assumption that it would be linear, which may not be true, but it gives an indication of how fast and how early the differences could show up. So for spectroscopy, for example, if I extend the rate of progression, you see that at age of onset, there are already large differences between controls and patients. And extended in the past, with linear changes, it would be around 8 years before onset that we start seeing the difference. There is another possibility that during development, the spine doesn't reach the same levels as controls. So instead of going here, it would be lower to start with. But I think this is very interesting because it could give information about the brain and the spine, actually the spine in this case, before onset, and give information on when to start treatment, once treatment becomes available. And in contrast, the spine cross-sectional area, if I do the same thing, extending the progression between V1, V2, and V3 toward the past, the crossing is much closer to onset. So that suggests that, for example, spectroscopy can detect changes before onset, and other modalities may not be able to do. So in summary, for this part, longitudinal changes can be observed in the cervical spine with all three modalities, morphometry, spectroscopy, and DTI. High effect size allows detection of changes in relatively small cohorts. Some of these changes likely occur before onset of symptoms. And so we think that MR could be very useful to serve as secondary endpoints in clinical trials in Friedreich's and in assessment of presymptomatic individuals. And we are very interested and excited to participate and bring these tools to clinical trials. And now, before I finish, I would like to show you some pilot data in dentate nucleus and in dorsal root ganglia. So these are preliminary data because we have only 3 controls and 3 patients. But I think it kind of expands what we could bring to clinical trials. And especially, it's interesting because dentate nucleus may be a target for treatment with gene therapy in several projects in development. And so the first one is dentate nucleus. So dentate nucleus is very rich in iron. But if we do T1 images, we don't have much contrast. We barely see the dentate nucleus. But if we use SWI and QSM, we see it much better. And for this work, we drew on several recent publications by Dagmar Timman's group that showed SWI and QSM in Friedreich's ataxia. And it turns out that Dagmar was in Minneapolis this summer, so we had a chance to talk about it. And we did a 3D gradient echo with echo time, 8 echoes, and SWI was processed online, and QSM was processed using a QSM MEDI toolbox from Cornell University. So these are 3 different images. The first one is a regular gradient echo image. The second one is the SWI image. And the third one is QSM. The QSM is not obtained immediately. It takes maybe 10 minutes, 10, 20 minutes to process. But you can see there is much better contrast with QSM than SWI, and especially if you want to determine the volume and do segmentation is much easier to do on that kind of images than on this one. So that's what we did. We developed tools to segment the dentate nucleus and calculate the volume. At this point, the data in 3 patients and 3 controls doesn't show any difference, but it's only a very small sample. The previous data published by Dagmar showed about 15% decrease in Friedreich's compared to controls in a more advanced cohort with FAR scores around 70. So it's still work in progress. And lastly, pilot data in dorsal root ganglia, again 3 Tesla, and we use a 3D DES double echo steady state sequence that provides a very good contrast of tissue to CSF. The CSF is almost completely suppressed, so we see the nerve roots very well. And the resolution was 0.8 millimeter isotropic. So this is an example here with lumbar dorsal root ganglia. You see the images are a little bit noisy, but we can see very well the dorsal root ganglia in the lumbar region. And again, here we did some segmentation to determine, so we didn't try to determine the volume because it's difficult to know exactly where the DRG starts. So we decided to do the diameter at the point where the DRG is the largest. So we took a perpendicular slice at the point where the DRG is the largest, and we calculated the diameter. And we found here on 3 subjects that the diameter was slightly lower in Friedreich than in controls. So that's encouraging. It's not statistically significant, but we think it could be useful to monitor treatment. So now we'd like to acquire data in more patients and controls. That's it. So I would like to thank first the FARA, who has been funding most of this project over the past 4 years, and Ataxia UK and GOFA provided funding for part of the first year of longitudinal scaling. And our work is also supported by QFA Foundation, Agilis for the dentate nucleus and DRG imaging, the Bob Allison Ataxia Research Center at the University of Minnesota, National Ataxia Foundation, NIH, and the Institute for Translational Neuroscience. Thank you.